Hello, microbiologists. Okay, we are going to start with some very exciting cell membrane stuff. Yeah, because remember, we're talking about staining, we're talking about cell membranes, we're talking about gram-positive, gram-negative, and peptidoglycan. That's where we left off in Chapter 3. Here's some very exciting videos about cell walls.
The cytoplasmic membrane of a prokaryotic cell is the main gateway for movement of molecules in and out of the cell. This membrane may also be called the cell membrane or plasma membrane. Cytoplasmic membranes in bacteria are comprised of a phospholipid bilayer. Each phospholipid contains a phosphate head located on the outer part of the bilayer, attached to a glycerol and 2-fatty acid, which face the core of the membrane. The phosphate is negatively charged and hydrophilic, while the tail portion of the phospholipids is hydrophobic. Thus, cytoplasmic membranes are very effective at blocking passage of many molecules, while other molecules are allowed to pass. Cytoplasmic membranes also contain two types of proteins. Integral proteins often span the entire bilayer and act as channels for transport of molecules that normally wouldn't be able to penetrate the bilayer. Peripheral proteins are loosely attached to the membrane on one side or the other. Some of these may be glycoproteins, which are proteins bound to a polysaccharide. These proteins may function as recognition sites, enzymes, or receptors.
That should mostly be a review from Gen Bio, yes. Okay, so again, membranes. What are we doing with them? They're for energy storage, they're for harvesting light, they're selectively permeable. But remember, in micro, we want death. We're trying to get in there and kill that little thing. So we want to know, how do we get into that membrane and kill it? They're naturally impermeable to most substance, so we want to create a substance that will get in there and kill them. The proteins do allow some cross-membrane activation and ways to get in. So again, we want to know what those are. We want to look at the concentration and electrical gradients so that we know how the bacteria moves things in and out. So here's another video.
With their hydrophobic cores and charged surfaces, cell membranes are very efficient at blocking passage of many molecules. Some molecules, such as water, dissolved oxygen, and simple alcohols, are able to pass through cell membranes freely by passive diffusion. However, charged molecules and large molecules cannot pass directly through cell membranes, and hydrophobic molecules tend to become stuck inside the hydrophobic interior of cell membranes. In order to allow these complex molecules to enter and exit the cell, organisms rely on two forms of membrane transport: passive transport and active transport. Passive transport occurs along an electrochemical gradient. It does not require the expenditure of energy in the form of ATP for transport. Active transport requires the expenditure of ATP in order to transport molecules across the membrane. Active transport and some forms of passive transport require the use of transport proteins, such as the integral protein seen here.
So again, that should be a review. Let's get out of all of these things and go back to the slides. Okay, so you're going to have an electrical gradient. You're trying to get things in and out of the cell membrane. And in this case, we're trying to get sort of antibiotics in. We're trying to really decide how each one of these microbes lives so that we can move things in and out of its little cellular wall, its little phospholipid bilayer. So you're going to have charged particles, less charged particles. Things are going to move from a higher concentration to a lower concentration, right? Remember, if I shove you all in a closet, that's a high concentration of people. If I open the door, you're just going to want to leave. I'm not going to have to push you out of the closet. If I open the door and there's a high concentration of people in that closet, you're like, no, I'm out. High concentration to low concentration. Everybody wants to be like evenly spaced, kind of. So passive transport, that's just the rules of diffusion. High concentration to low concentration. These things look about in equilibrium, right? These things it looks like there's a higher concentration here, so they'll diffuse in and out. Osmosis involves a very specifically water and a semi-permeable membrane. And water, remember, likes to rush in and dilute things. So if there's a high concentration here, this water is going to automatically want to rush in and dilute things, because that's what water does. Okay, bacterial cytoplasmic membranes. So now we're talking about bacteria. Typically passive, the passive processes, so these don't require any energy. This is just diffusion. This is like high concentration to low concentration. There's not even a wall there. Like you open something stinky and it just goes for diffusion. Just in the air, in water, you drop. Maybe if I dropped some food coloring in water, it would just diffuse, right? That's just diffusion. Facilitated diffusion is something sort of taking it with them. One chemical is sort of making that process go faster or helping it along. And osmosis involves water and a semi-permeable membrane. Okay. Again, passive processes, extracellular fluid, facilitated diffusion, several types of chemicals through a non-specific channel. That's called facilitated diffusion because what's facilitating that here is this channel. Or you can have facilitated diffusion through a specific chemical channel where you have to bind a certain way to get through. Or this one is just osmosis. I can move myself here, maybe up here. And this is just osmosis through a semi-permeable membrane. If you remember this from any kind of intro bio class, this is the diffusion of water across the semi-permeable membrane. Okay, we have more over here. Where does the water want to go? The water wants to dilute. The water wants to dilute this, right? So water is going to rush over here to try and dilute it. Does this remember this? Yes? Okay, so we have hypertonic, isotonic, hypotonic. So right here, we have the same amount on the inside as the outside, so this is going to be isotonic. Here, we've got a bunch more out here, so the water is going to rush out. Remember, water's going to dilute things. So the water's going to rush out of here, and it's going to shrivel up this cell. It's not great, right? If that's your blood cell, you don't want it shriveled up. Nobody wants that. That's like, no, 1 out of 10, no thank you. Again, process in a cell wall. Isotonic, the cell is happy. Everyone's happy. This, not as happy, right? If you have a hard cell wall, that's fine. You're not going to notice it shriveling, but this is shriveled on the inside. This would be, you can see it in plants when it's trying to dilute things. Could be your blood cell. You don't want that as your blood cell. All of the water is trying to get out to dilute something in your blood. That's not great. Conversely, you got a bunch of solute inside your cell, and water is going to try and rush in to dilute that. Same thing here. You got a bunch of something inside the cell, and water is going to rush in to try and dilute that. This is what happens when morons give people a bag of just pure DI water right into your blood instead of the actual correct type of with the amount of solutes that should be in there, electrolytes and specifically sodium. That's why IV fluid isn't just, a bag of water. Because if you put a bag of water in there, what's going to happen? You're going to have water out here, and this is going to be a different concentration, so the water's going to rush in, and it's going to burst your blood cell. So don't give people an IV of water. Don't do that. Again, different kinds of passive transport. Active processes, this is coming specifically across that semi-permeable membrane. So you have different kinds. You have uniports, antiports. Basically, these are just going to come in and see how you have ADP, ATP. This is going to use energy. Your body is going to try and limit this as much as possible because it doesn't want to waste energy just getting things across the semi-permeable membrane unless it absolutely has to. But there are certain things that, carbon, phosphorus, these things have to come across. Here's a little transport process if you'd like to know how things get through. Active transport, usually sodium-potassium pumps, calcium channels, hydrogen ion channels, again, chlorine channels. These things, they're very specific in your body and you need very, very specific amounts of them. So they're usually ATP dependent and very touchy, if you will. So that's, we need to know that about us, but we also need to know that about microbes so we can potentially will they die in a salty environment? Maybe, right? Maybe we can just use osmosis and diffusion to kill them with salt or chlorine or something like that. Okay, next video.